Everyone, and welcome to our video on mutations. So far in this module, we've talked about gene expression, which is the process of taking DNA and using it to make RNA and using that RNA to build the proteins that the cell needs. Well, remember, y'all, that DNA is replicated before a cell divides, and that DNA replication is essential because before a cell divides into two daughter cells, that DNA needs to be replicated quickly and efficiently so that the new daughter cell is able to have a copy of DNA. And remember, bacterial cells, a lot of them divide within 20 to 40 minutes. So DNA replication happens very quickly. There's a lot of enzymes involved in that process. And like anything, accidents happen. And so when there's an accident and the wrong nucleotide gets incorporated into a piece of DNA, that is called a mutation. And what I really want to emphasize right now is that mutations are happening at the DNA level. But because of transcription and translation, any mutation that happens in the DNA is going to affect the RNA and possibly the protein that gets produced. Okay? But mutations actually occur at the DNA level. Okay? So let's define the types of mutations that we may talk about. Now, we are going to be looking at silent, missense, nonsense, and frameshift mutations. Okay? And so a silent mutation is when the change in the DNA sequence doesn't affect the amino acid that gets made. Because remember, y'all, the genetic code is redundant. There are multiple codons that code for the same amino acid, like we just saw in the last video, the gene expression practice problem video. And so when the DNA mutation doesn't affect the amino acid produced, doesn't affect the protein at all, we call that a silent mutation because really the cell isn't impacted anyway. Think of it as no harm, no foul. But sometimes there is a problem. So when there's a mutation in the DNA and it leads to a different amino acid being incorporated, so the wrong one, we call that a missense. And I think of that as a mistake. Okay, so the missense, this is going to be our mistake. All right, when the mutation leads to a premature stop codon, meaning translation stops, the protein stops being made, then we call that a nonsense mutation because the nonsense mutations are not tolerated. Because think about what happens if translation shuts down too soon. Well, then the entire protein's not going to be made, which means it can't possibly fold into its correct 3D structure, so it's not going to be able to do its job. Especially in prokaryotes, most proteins are absolutely essential. So having a protein not work is going to be very, very problematic and can usually be lethal. Okay. The last type of mutation is when there's kind of like a confusion happening in the DNA replication process and an extra nucleotide gets added or gets left out. And so this is called a frame shift because the reading frame is shifted. What does that mean? So remember that we look at the RNA, we read it in sets of 3, right? I'm just making things up at the moment. Here we go. Well, sometimes during the DNA replication process, an extra nucleotide gets added. Well, that means during transcription, there's going to be an extra nucleotide added because RNA polymerase doesn't know what the DNA should be. It just uses whatever DNA it's given to build that RNA. So if the DNA has an extra nucleotide, it's going to throw in an extra nucleotide. Well, what does that do? Well, let's highlight in yellow. Let's get my colors going. what the original sequence was, right? So remember, it was AUG, CCC, and then AAA. But now, y'all, because there was a mistake at the DNA level, right, that added in a nucleotide, in this example, AUG, the first codon, it's fine, no problem. But now look what happens. Because of that added nucleotide, oops, the second codon is now ACC. Then the third one becomes CAA. And you can imagine as we move down, right, now all of the codons are different. Our reading frame shifted. The reading frame are the sets of three. Our sets of three have now shifted, and we're essentially going to build an entirely different protein, right? Because now all of our codons are different. So let's go ahead and look at a quick example. And I believe my example is on this side of the page. So I'm going to just erase some of this real quick. All right. So let's take a look. So here is my little DNA sequence. And we know it's DNA because there's a thymine in there. So let's pretend that's my DNA sequence. Well, normally the RNA that's going to be made, oops, oh, golly gee, y'all, sorry. All right. I forgot what I had put on my own slide. I'm sorry, y'all. So if my DNA is normally ATG, then what RNA should be made from that? Okay, so I want you guys to actually tell me real quick. So I recommend you stop and pause the video and go ahead and write that down. All right, so remember, we're thinking of base pairing. So A pairs with U and RNA, because remember there's no thymines. T pairs with A, and G pairs with C. Okay? And if we look at our codon wheel, which I apologize, I did not put on this slide, UAC gives us the amino acid tyrosine. Okay, so that's what should have been. Well, if we say that there's a mutation, and instead of the thymine getting incorporated, instead an adenine was added, that means that we're going to have a uracil added in our RNA instead. And then if we look at our codon wheel or codon table, UUC gives us phenylalanine. So which type of mutation is this? Well, it's a mistake, right? We still made an amino acid. We just made the wrong one. So this would be a missense. It's a mistake. Okay? Because remember, y'all, a silent mutation would mean that the amino acid didn't change. Well, this one did. A nonsense mutation would be a premature stop codon, but we didn't get that. We got another amino acid. And our reading frame, our set of three did not change, right? Just we had the wrong nucleotide put in there. Okay? All right, so now let's go ahead and do another apply what you know. And so here is the template DNA strand. So I want you to go ahead and pause and try to work through this. What RNA should have been made had the DNA not been mutated? What amino acid would have been made? And then I want you to play the video. I'll check your answer, and then I'm going to introduce the mutation. Okay? All right, here we go. So remember, to figure out the RNA sequence, we are going to do our base pairing. So G pairs with C, A pairs with U in RNA, C pairs with G. Now we're going to look at our codon wheel, and we're going to start with the C here in the middle. We're going to work our way out to U and G. That gives us leucine, L-E-U. All right, how'd you do? I bet you did awesome. All right, so now if there's an error, I'm going to go ahead and change my pen color for this one. If there was a mistake when the DNA was replicated, and now the DNA sequence reads CAC, what's going to be the impact of that? Well, if the DNA reads CAC, that means the RNA is going to be GUG. And if we go to our codon wheel, GUG is going to be valine. So this is another example of a missense mutation. So let's do another one. Again, I recommend you pause it and work through it on your own. So if the template DNA is ACG, then the RNA would be UGC. Sorry, I know that looks like an A. That's a U. If we go to our codon wheel, UGC is cysteine. All right. Now let's go ahead and check out what our mutation is going to be. All right. So a mutation happened during the DNA replication process, and instead of the ACG, the sequence is now ACT. Well, during transcription, RNA polymerase doesn't know that that's wrong, so it's just going to do its base pairing. So it's going to read UGA. Let's see what that gives us. You go to the codon wheel. We have UGA. Uh-oh. It's a premature stop codon, so no amino acid is going to be incorporated. Translation comes to a halt. This cannot be tolerated. This is considered a nonsense mutation. Okay, nonsense mutation. All right, let's look at another one. So the template DNA should have been TTT, which means the RNA would be AAA. And if we use our codon wheel, that gives us the amino acid lysine. All right, let's go ahead and check out what our mutation is here. And please know the mutation can happen at any of these positions, okay? So if in this case the template DNA is now TTC, that means the RNA is going to be AAG. And if we use the codon wheel, AAG gives us lysine. Hey, no harm, no foul. The same thing is made. The cell has no idea there was ever a problem. This is our silent mutation. Okay, because the same amino acid still gets incorporated even though there was a mutation. And remember, this is because of the redundancy of the genetic code. Multiple codons, and you see it happen in a lot of places, right? You see it here, you see it here, other places too, right? This one has two. And so we say the redundancy is that multiple codons give the same amino acid. And it tends to be, if you notice, in the third location, okay, that third nucleotide. And because of this redundancy with the multiple codons giving the same amino acid, it helps increase the likelihood that things are going to be okay if there's a mutation, because there's a chance that it's going to be a silent mutation and the same protein's going to be made, meaning it's still going to fold correctly. It's going to be able to do its job. The cell will be fine. Okay, so redundancy helps make silent mutations okay. All right. That is going to be the end of this video. As always, y'all, let me know if you have any questions. Bye, y'all.